the basic idea of the underlying model that I'm trying to kind of promote is this notion that scientific work is a cyclic process. You start off with scientific knowledge that is contained within the scientist's head most of the time. They then analyze and think about the work that they're trying to do. They come up with new ideas, new theories, and they come up with good scientific questions that are relevant and important and actually make a difference in the subject they're asking. In order for this to be scientific work, those questions need to be addressable in an experimental way. So you have to actually have experimental designs and protocols that test and execute the processes for that. And then once you actually have your scientific protocol, you execute an experimental design. You actually populate the data. You acquire data, and you gather the data. You get hold of it, and then you interpret it to add to your scientific knowledge. And you kind of and so the whole purpose of what we're trying to do is to figure out how we can go around this cycle more quickly. How can we build tools and systems to accelerate the process around this so-called knowledge turn? And the kind of key question is, how do you make scientific discoveries? What is the crucial element of making scientific discoveries? Well, in some ways, we want to be able to analyze existing knowledge that comes up with important questions that then are testable scientifically. If you think about it, this is the absolute crux of what science is really revolving around. And it comes down to, how do you find the right questions? Okay, how do you actually do that? Now, if you're someone who can weigh the universe on the back of an envelope, this is not really a problem. You're someone like Einstein, you can figure this stuff out for yourself without too much difficulty. You could also be someone like Rosalind Franklin, who is the kind of poster child of someone who did really serious, diligent work, really solved a problem that was important, and unfortunately wasn't actually recognized for it. She didn't get the Nobel Prize for her work because they don't offer those things posthumously. But instead, these guys, Watson and Crick, were able to come up with exactly the right question at exactly the right time and solve the problem in a really kind of innovative and creative and interesting way. Okay? And in a way, again, this is what computers can do. Perhaps computers and computational technology and knowledge engineering technology can help us find those questions and answer them in an appropriate way, which is kind of cool. So a notion that underlies all of this work is this, that is actually interestingly missing from most people's accounts within bioinformatics of how we should address these kind of questions, is this notion of scientific paradigms. Now, I don't know if you guys are familiar with the work of Thomas Kuhn, but in 1962 he wrote the seminal work called The Structure of Scientific Revolutions, in which he talks about paradigms as being some accepted examples of scientific practice, examples which include laws, applications, and instrumentation together to provide models from which spring coherent traditions of scientific research. And so, you know, we have this kind of underlying idea of what a paradigm is, but what's really interesting is, how do we actually transform the knowledge within a paradigm shift, within the framework of a paradigm shift? Now, so let's just go through a kind of simple example to illustrate what we mean by that. A paradigm shift from elements to the elements, to the chemical elements. And in fact, this is a kind of illustrative example. So typically what happens when people come across a new phenomena, such as fire, they try and kind of categorize the different types of fire that they have. They do stamp collecting. They kind of figure out, okay, this type of fire is very green. This kind of fire is blue. This kind of fire is very bright, that kind of thing. And of course, a lot of science actually falls into this category at the moment. People come across phenomena that they don't understand very well, and they try and kind of explore it and figure out how the different phenomena work. And of course, when they do that, scientists typically will make up a theory, and they'll postulate some mechanism that may or not be accurate. So in the case of fire, of course, the original idea was that fire is a stuff called phlogiston that has some kind of properties like a fluid and such like. But of course, it was the wrong theory. It didn't work. So what happened there is that the paradigm kind of got stuck. It got in a loop. The predictions that were being made weren't accurate. They weren't, the predictions that were being made weren't consistent with the data that was being gathered, until, of course, Lavoisier came along with the brilliant idea that, in fact, the correct mechanism was that the process of combustion involves oxygen and carbon and produces water and releases energy, which is what fire is all about. Now, of course, what's interesting about that is that it then formed, this discovery then formed the basis of a whole theory, the theory of modern chemistry. And once they were able to do that, then they were able to export the theory as a kind of structured reasoning framework, something that scientists in other fields could use for their own purposes. And of course, the periodic table is a great example of that kind of thing. And that's actually the kind of thing that we're trying to address within the knowledge engineering work that I'm going to be talking about today. And of course, once you've actually got a theoretical framework which you can operate within and work within, then you can develop new technology. You can do anything you like. You can actually build combustion engines. You can cure disease. You can do all sorts of stuff. So one of the important distinctions that I'd like to make about this cycle, this cyclic process, is this distinction between interpretations on the left-hand side and observations on the right. Now, what's interesting about this is that the people who are kind of great at dealing with these different processes are different people, right? On the left, you have Professor Honeydew, who is an expert in the field. He's steeped in the knowledge acquired over many generations. He knows the details of exactly how to interpret everything, and he's the guy who comes up with new experiments. Whereas on the other hand, you have Beaker on the other side, who is able to, who's capable of reproducing the experimental data. He's able to execute a well-defined scientific protocol, but he doesn't necessarily understand why. And that's okay. That's actually in our case is a good thing. Now, the distinctions of interpretations versus observations within this context is that the interpretations are semantically complicated, whereas the observations involve only really data semantics, which is actually quite tractable. On the left-hand side, you need a human in the loop. On the right-hand side, you don't. You can automate stuff. On the left-hand side, you have knowledge that has to be specific to the individual paradigm. Like if you're an expert in neuroanatomy and you know how the structure of the brain is functioned, when you're trying to design and develop experiments within that field, you're going to be pulling on that kind of theory, whereas a neurophysiologist doesn't understand the details that you would be using. Okay, whereas if you're just trying to process neuroanatomical data, you don't need to know that stuff. It's just data. It's nice and coherent. And so the idea is that the way in which we look at these two different situations has to be a little different. In order to make interpretations tractable, you want a scoped representation of the paradigm that allows you to actually kind of reason within the structure of that paradigm effectively. Whereas if you're working with observations, as long as you keep it restricted to data and measurements, then it's tractable. You can calculate all sorts of statistics and statistical effects quite happily. Okay, so that's an important distinction to make. And because we are trying to keep it simple, and because we're trying to build this within a computational framework, we're going to tackle first the observations, and we're going to address that kind of question. So the work that we do is based around this kind of notion called knowledge engineering from experimental design. I'll get to it in a second. The idea is that this is a slide that you would probably see in scientific talks about biology all over the place. At the top is this scientific statement. That's just here. That would be an interpretive statement saying something like rats eat cheese when they're hungry or something fairly straightforward. And it's usually based upon data, and the data is going to be based upon measurements, i.e. of a dependent variable, and the settings that you set within your experiment before the experiment starts to test your effects as independent variables across the board. So here, the kind of important criteria that you're looking for here is there is a difference between conditions D and E, and it's illustrated by the measurements of the dependent variable. So that's actually how scientists think about their data most of the time. This is the underlying conceptual model that they use to design and build experiments and to actually do their technical work. And what we've done is we've tried to extrapolate away from that to provide a simplified framework for scientists to be able to describe what they're up to using these representational elements, using processes, material entities, parameters, constants, measurements, branch points, and fork points. And given this kind of vocabulary of conceptual structures, it's actually not too difficult to get scientists to describe their protocol and to draw out their protocol in the way that I've just shown you. Okay, so and actually I gave a talk at a a neuroscience undergraduate lecture, and I showed them this slide, and I asked them to put their hands up, and I said, how many of you feel comfortable that you could do that? And most of them did, which I was like, great, that's awesome. Okay, so now what's interesting about this, if we're trying to extract the relationships between dependent variables and independent variables, measurements and parameters, we get that for free from this kind of diagram, because in fact, we're able to trace back through the structure of the measurements to see which are the variables that actually parameterize the process upon which the measurements were taken. So in fact, you can see that measurement 1 is dependent upon parameter 1 and parameter 3, but not dependent upon parameter 2 because it lies on a different arc of the protocol. Okay? Now that gives us our relationships between independent dependent variables that can form the basis of a data representation, which is kind of cool. We actually have a system that we've built called NeuroScholar that in fact provides a drawing palette that scientists can use to draw out the protocol that they're actually using. So here, this is a very simple experiment. You take a mouse, you measure its blood pressure. Nothing complicated about that. And the system allows you to describe the various different variables at different stages with different data types and such like. Very happily, we were able to publish this as a paper. Can I get this? For some reason, here we go. So you see on the right-hand side a reference to a paper in BMC Bioinformatics, and I'd just like to point out that the first author of this is sitting right here. This is Tom Russ, thank you, who was working with me on this work previously. So, and this is a highly cited paper within the community, so we actually got some impact. People were reading it. It was quite good to see. Okay, so so having kind of set up the structure of this knowledge representation where we're trying to capture the primary measurements by using these provenance relationships through the protocol, we then confront the unfortunate fact that data processing transforms data. Well, obviously, right? That's not surprising. But if we're able to actually capture that as well by seeing, if you look at this representation on the graph, the idea is this is an experimental protocol that consists of taking the MMSE questionnaire, which is the mini mental state exam, which is a test of cognitive ability that people will take if they're suffering from Alzheimer's. And you can actually see that the data structure is relatively simple. You have your score, followed by your ID number for the subject, and then the group that you're in. And if you want to calculate the mean values of this specific measurement, which is an important step to take, you then have to, the process of calculating the mean will average out the IDs. No longer the individuals will be measured there. So by mapping the inputs and outputs of each individual data processing, we can construct a representation for actually that will support a knowledge representation for the following elements: for primary measurements, for mean values, for statistical effects between groups, which we're showing at the bottom here for the MMSE Z-score, and of course correlations, which are not shown here. So the idea is that all of the kind of statistical relationships that you might see in a paper, if we adhere to this kind of modeling approach, we should be able to capture and represent quite cleanly and easily within this approach. Now, how expressive is this? How good is this as a methodology? Now, you can see this is a paper that's concerned, this is a data figure from a paper concerned with looking at vaccine responses to an HIV vaccine. And what you're looking at here is a slide that shows kind of a horrible data slide, right? But this is not completely atypical of the kind of things you see in scientific papers. The thing that you're looking at is that the black dots are supposed to be higher than the white dots. The black dot shows an immune response under conditions of the vaccine, and the white are a control, okay? Now, if you draw out the entire protocol using the kind of conceptual framework that we described previously, you get this complex representation. And in fact, the measurement that we're looking at is shown by the number 5, the circled measurement in the diagram. And if you trace back through the protocol, through every single individual parameter that indexes that measurement, they correspond exactly to the measurements shown in they correspond exactly to the axes of this diagram shown here, which is very encouraging. Okay, so we think that given the complexity of this data, so we were actually able to go through this paper and curate all of the data points, every individual data points from this paper into a database representation, which is actually really, really cool, because that's actually very, very difficult to do using other database methodologies and data modeling approaches. Okay, so that's the kind of core, that's some of our core technology, that's the kind of approach that we're trying to take. Now, we're also trying to think about, okay, how do we apply this approach? How do we apply this approach across a whole field? Now, given the complexity of the different types of research that people do, we don't know the number of paradigms that people are working within. There are all sorts of different ways in which people are doing the data analysis. And three examples of the kind of data formats that people use are shown here: human neuroimaging, protein-protein interactions, gene expression. All of them involve different types of data, but they also involve different kind of hidden, intrinsic methodologies and assumptions that people make when they're driving the kind of research that they're doing. Okay, so we want to know how do these different, well, let's ask the question, how do we figure out how many paradigms there are? Well, you guys are all very familiar with topic modeling as a methodology. Maybe we could take all of the text within a specific corpus of scientific papers and try and automatically discover the kinds of paradigms that are within. This is an explanatory slide that I think, after talking to Brian, is probably completely unnecessary. The idea is, obviously, topic modeling involves taking documents like this. This is just an abstract from the Society for Neuroscience meeting a few years ago. And what topic modeling allows you to do is to take, is to identify words that tend to reoccur together in the same document again and again and again. So if you're working on Parkinson's disease, for example, you'll see these kinds of words like nigral, dopaminergic, substantia nigra, all co-occurring within the same document. And so by doing topic modeling, you can then model the structure of the document itself as a mixture of different topics distributed over the entire corpus. And that gives you a framework within which you can then perform statistical analyses on the papers that much more easily. And what we did was we wanted to kind of provide a graphical representation of the layout of a subject that scientists could eyeball and examine. And so what we did was we took a literature corpus, we generated a topic model from it, we then calculate document similarity scores based upon comparing the different topics themselves for each document. And then we actually embedded this in a Google Maps application to provide a kind of a layout that you can then zoom into and look at and examine. And this is actually quite old work. We did this back in 2007. And this is a cluster map of the 2006 Society for Neuroscience meeting, which is actually kind of interesting semantically as well. This is a conference that about 30,000 different scientists come to every year. It is the primary main conference for all of neuroscientists across the world. And in a way, it's kind of like a snapshot of the state of the art at that point, which is kind of interesting. And all we're showing here is this layout that you can kind of zoom into. The nodes are colored by the different themes that they were manually classified into within the conference description. And so you can see that it forms these clusters. And it's an interesting idea to think, can these clusters of different nodes be used as the basis of defining or investigating or looking for paradigms? And this is work that's still ongoing. We haven't really developed this a great deal, but this is an example of the kind of work that Google could in fact do, obviously at scale, rather than dealing with the 11,000 or 12,000 documents that I'm looking at here. Google could certainly examine, for example, the 20 million documents that occurred within PubMed. These are things that you know, I'm trying to kind of throw into your space as possible avenues and ideas for research that you might be interested in taking up. Okay, another example of this is the NIHmaps.org project, which was work that which was derived from our original idea. We worked with Ned Talley and with a company called Chalk Labs to develop this application. And actually, I definitely want to make it very clear that we had no real part in the development work here. This is all Chalk Labs development work, but nonetheless is kind of inspired by our original idea. And what you're looking at here is a representation of the 80,000 or so grants that were actually funded last year by NIH across the entire, by the whole US government. And what we're able to do is query and search for grants that contain 30% or more of their words classified as Parkinson's disease relevant words. And you can see that you zoom in and you actually get a small cluster of papers that are concerned with deep brain stimulation, which is a specific methodology. So in theory, everything within this kind of cluster may be relevant to the same kind of thing. That could define a paradigm. Kind of interesting. And it's also relevant because these guys are all funded, right? So it's not just a simple theoretical question about, okay, who's doing this work? These guys are all actively processing and developing the same kind of ideas. This is kind of a little hard to see, I apologize, but this is a breakdown of the different categories of topics that occur within this corpus, and they cover the gamut. And of course, this is quite interesting because then you can actually see what the literature is telling you as are the most prominent words that are being used by scientists, rather than ontologies that are defined by informaticists. These are the actual words that are in the documents. So that's another interesting avenue of research that we could all kind of think about and work on. So now I want to talk about something evil, dark data, dun, 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 right? And of course, if you've not heard the term before, dark data is an allusion to the concept of dark matter, which is data that matter that exists, but matter that we can't see. It's kind of out there somewhere. We know it's out there. We have no idea what it is, which is kind of cool, but it's present. Now, when you're thinking about data and dark data, this is data that exists. We know that it's in people's labs, it's in people's notebooks, it's in people's filing cabinets, in proprietary file formats, in all these different places, but we can't get hold of it and we can't see it. And the fact is that so much data is kind of hidden away, and the infrastructure that we currently work within doesn't actually promote the sharing of data very effectively. It does so a little bit, but not very effectively. It has a big impact. Last month, I went to a meeting in Oxford where people were talking about scientific integrity and scientific rigor and the whole process of data sharing. And this guy, Chas Bountra, kind of stood up and talked for about 15 minutes without any without any slides at all. And he gave this wonderful impassioned speech about how destructive the process of concealing data from each other is within the pharmaceutical industry. And so he heads the Oxford division of the Structural Genomics Consortium, which tries to provide a pre-competitive space for drug discovery, where and he's actually getting a lot of really supportive money from pharmaceutical companies who are willing to chip in to try and basically support data sharing at the stage before which drugs become competitive. So there's all sorts of preliminary trials that people want to do to make sure that molecules are viable targets for actually developing drugs for. And he's trying to kind of develop the idea of being able to save some money for doing this. And so obviously the quote is, it's harder to get a working treatment for Alzheimer's disease than putting a man on Mars. The reason he says this is because according to his estimates, as far as I remember, I could be wrong about this, he said that it costs $30 billion or 30 billion pounds, I can't remember which one it was, for all the different research that's gone into trying to understand Alzheimer's disease, and they still don't have a working drug. Like all of the stage 2 pharmaceutical trials that have taken place have all failed, which, if you think about it, is horrifying. Okay, we should be able to do better than that.
There were 2 recent failures reported just a couple of days ago.
Really?
Yeah.
Specifically for Alzheimer's disease?
Yes.
Okay, so there are some more, there are recent failures in the last couple of days for Alzheimer's disease and drug development. So, okay, this is a very topical question as well, a very serious one. And so, you know, the people in the knowledge engineering community and the semantic web community have put together the linked open data cloud. It's a little blurry, I'm afraid. I'm sorry, you can't see it very well. And within that space, there are a number of different projects in the life sciences. And essentially what this is all about is these guys take these large online open databases and they dump them into RDF and they look for ways in which you can make connections between them, which I think is a very, very valuable starting point to demonstrate how data can be linked and data can be made interoperable in a very open way. However, having said that, the representation that they use doesn't make any distinctions of the kind that I just described at the beginning of the talk. If there are interpretations and observational data, they tend to be mushed together, and you need to know the underlying schema of the database to be able to make sense of them. So in fact, even though, and of course the different data sets that are contained within the linked open data cloud are all basically representing the data that's contained within the database as it stands. So you have all of the problems of linking together terminology and trying to understand how the different data schemas are representing the data and such like. So we haven't solved some of the major conceptual problems about how this kind of representation should be constructed and should be framed in a paradigm-specific way that would actually allow us to reason over and build representations for specific scientific problems. But I think that this is a good start, and this is an interesting way. This is kind of something. This is the way things should be in the longer term. Like this is a framework within which we could publish our data, and actually this could make a difference in the longer term. At the moment, this hasn't necessarily really proven to be a game-changing technology, but nonetheless, the promise is there at some point in the future. And now I'd like to also mention an approach that's being put forward by Baron Mons and an extended community of people, which is called nanopublications. So the idea is that you can find a representation in RDF that contains a named graph that stores a scientific assertion or a scientific data set, some form of representation that could be fairly general purpose and could be lots of different types of format that you can include there. But you have a standard representation for the provenance and for the supporting data that allows you to link together different data types. And I think this is a really valuable and interesting idea, and is actually a way in which we can try and think about transforming the way in which scientific data is published and shared as part of the whole publishing cycle. And some examples of this is you could have a data point linked to a paper citation, you could have a representation of a statistical effect, or you could have an interpretation based upon K-fed data. And these are all obviously conceptual ideas. We haven't got worked examples of nanopublications to show you yet, but watch this space. This is something that we want to work on. Okay, so having talked about this and having talked about the underlying K-fed representation and all of these various different other components, I wanted to kind of bring it home by talking about a real scientific problem, and actually talking about how one might be able to encapsulate and really represent a specific scientific paradigm in a coherent way. Okay, so talking about biomarkers, who here has heard of biomarkers as a representation? One guy, one person. Great, thank you. Okay, so the idea is, and actually biomarkers is a very well understood methodology within biology. The idea is that given an individual in some kind of disease state over time, actually, is this okay? Okay, good. Great. So in some kind of disease state over time, a person's disease state will change from normal, preclinical, mild clinical to severe clinical in a way that allows them to, and you can actually track that over time. Now, biomarkers, the idea is that you have various different types of measurement that you make on that individual over time that actually allows you to track various different aspects of the disease. And so here's an example of a numeric biomarker. Of course, this is all just cartoon stuff, or a binary biomarker that shows a state of either being off or on, and imaging biomarkers that actually indicate, the MRI images that reflect a person's structure of the brain. And the idea is that essentially this whole, the way in which the biologists think about this is they're looking for an indicator test. They want to find something that indicates the transition from a normal to a preclinical state. And they want to find a test that you can deliver as a blood test or a questionnaire or something that's indicative that this person may have a disease of some kind. Okay? And so what's interesting about that is that if you think about this, you have an individual whose state changes in discrete stages over time, and you have a whole bunch of features that indicate measurements of some aspect of that individual's in measurable quantities over time as well. Now, doesn't that sound like a hidden Markov model to you? Isn't that something that computer computer science can actually address in a very tractable and easy and straightforward way. Well, not necessarily easy and straightforward, because the features you're looking at are very complicated, but nonetheless, it actually falls within the domain of computer science. You can address that kind of question. So the goal, in a way, of what we're trying to do by shaping and representing the data that we get access to is to make it available and accessible to people like you, to computer scientists, to experts who are really, really, really good at data analysis at scale, and can actually address these kind of questions. Okay, so let's then see where this is applicable to. Maybe, there are these two projects called the Alzheimer's Disease Neuroimaging Initiative, or ADNI, and a more recent attempt to analyze and look at Parkinson's disease data in the same way, called the PPMI, the Parkinson's Progressive Markers Initiative. Now, these two projects are kind of interesting. They're large-scale multi-site projects that involve clinical sites across the world in lots and lots of different places, where they have to coordinate very carefully the data acquisition and the representation of different data from different places. They're actually exemplary, really, really good at that. And the idea is that they simply capture and track 200 patients in a disease group and a non-disease group over time, over a period of 5 years. And they capture imaging data, they capture blood, they actually do spinal taps and take cerebrospinal fluid and freeze it so that people can do subsidiary, secondary backup studies to look for specific things within the tissue. They do questionnaires, they do clinical assays, they look for various different, any kind of clues that they can find that may suggest a transition and a progression of the disease. And importantly, they actually provide access to all the data, and they provide access to the samples. If you have to go through a rigorous application procedure to get access to this stuff, but it's available for anybody. So these guys are really setting the stage and pushing these things forward, and really kind of having a big impact. And of course, surprisingly enough, or rather not surprisingly enough, these projects are really successful. They actually are, there's a paper out by the guys who run ADNI. ADNI is now in its second phase. It's a more international consortium now. It's globalizing. It's scaling itself up quite well. And they published a paper that shows something like 200 papers were published around this individual study originally. And of course, the impact they have on secondary studies is even greater. So the
data is available. The data
exactly.
It's not like, hey, I'm not going to show it to everybody because I want to publish first.
Yeah, exactly. They're saying, look, we want you to publish, we want you to leverage this stuff, we want you to succeed. Okay, so by taking this kind of process, the people who developed ADNI really pioneered the whole kind of data sharing framework. And they did so in a way that's actually kind of interesting, because 200 patients per group is not massively scalable. It's not at a level of, 10 million, the population of China or something like that. It really, but nonetheless, it's actually able to solve these problems and actually really make some progress about trying to detect Alzheimer's disease in people. The PPMI project is a little younger. It's only been around for a short period of time. It's a large-scale international consortium of 24 separate clinical groups, all of whom are working together. It's sponsored by the Michael J. Fox Foundation, and it actually is kind of and this is the main presentation of from the website, the PPMI, and so they have a whole complex governance structure where they have different working groups developing and building these things. And of course, what's interesting about this too is that all the researchers who are working on this problem are working on their own problems in their labs. So you have these top researchers from these different clinical labs all contributing to this centralized project, but that's not all they're doing. And so the community built by this project itself is able to kind of share data in a knock-on effect as well. So I think that's something that we kind of underestimate the impact of, and it's actually one of the reasons why I'm very excited to try and work with these guys. Now, what's interesting about this as well is they're sharing the data, but the data is all shared as CSV files, text-readable CSV files. Usually with there's a data dictionary that you can look stuff up in, and all of the data descriptions are written down in PDF files so that you can actually learn and understand the data if you're an expert and if you take the time. But for a machine-readable approach, using semantic web and knowledge engineering, it's actually not enough. We need to innovate. We need to bring our technology into this domain, and we need to be able to kind of actually model the process using the kind of tools that we've done. So we've started this work. This is a K-Fed knowledge engineering from experimental design model of PPMI. This is completely unreadable, so let's make it a little bit bigger. The idea is that you have an individual participant, and then you have a loop structure here that actually provides a representation of an individual visit. And a visit is basically where they do all the work. And then you have a whole bunch of assays leading off into the distance over there for the various different types of measurements that people can make. And the work, we've only just started work on this. I basically put this model together last Saturday, but we're able to actually build a complete representation of all of the different types of variables that people do at this level. We haven't done any of the knock-on studies that people do on top of things like blood samples, urine samples, and CSF samples, but nonetheless we're able to capture, for example, the structure of the data that's being measured and represented within the original data set. So I'm very encouraged about this, and I think that this actually provides us with a framework that then we could publish nano-publications based upon PPMI data, assuming, of course, that we get permission to do so. We have to apply through their internal processes to make sure that that's okay. But then, excitingly enough, what I'd like to do is to present all of this data in a machine-readable form that people like you can then process. And so we could actually present this as a framed data set that anyone could have a crack at with respect to using hidden Markov models or CRFs or any kind of data modeling kind of approach that we have. So in a way, again, this illustrates the role that I think the work that we're doing actually provides by being a bridge by being a framework for capturing the semantics of the biomedical processing in enough detail so that we're able to kind of actually capture the underlying data and representation in order to solve problems, but not so much detail that we start trying to invoke all the knowledge that these guys carry around in their heads, because there's no way we can do that. But then we want to package and present it to you guys. So in a way, that then exposes all of the data for all the sorts of kind of large-scale, complex and interesting machine learning kinds of analysis that are in your toolbox. So that's kind of part of the mission of what we're trying to do and how we can actively pursue this and actually make some progress and maybe even cure some of these diseases. Okay, so going a little bit further, that's the main body of the talk, but looking further on off into the future, a couple of ideas for you to consider. So first of all, this is a slide that describes, this is a slide that describes this is a famous slide from a neuroscience textbook that illustrates the different levels of organization of the nervous system. You have genetics, you have various different proteins in channels in the cell surfaces. You have synapses, microcircuits, neurons, local circuits, whole brain structures, then behavior. And of course, a disease like Parkinson's disease actually affects all levels of this framework. You have to be able to look at the research at all different levels. And in fact, the kinds of questions, if we're interested in studying not just the correlative effects of these different symptoms, which is what biomarkers is really doing, if we want to start digging into the causes of how the disease works, then we have to start looking at these various different elements. So LRRK2 is a gene, and is in fact, I think the gene that Sergey Brin has a mutation for, that is part of the reason why he's actually interested in studying Parkinson's disease and trying to find a cure himself. There is also work done. Part of the reason why Parkinson's disease causes damage is it's based upon aggregations of these various different malformed proteins that grow and cause problems and basically kill off cells. We don't know how, we don't know why, but the processes that underlie that is somehow related to the way in which the proteins fold. Again, we don't understand exactly how that works. There's a whole, that itself is a paradigm. People work within that field their entire lives trying to understand that whole process. Then the process of, let me get this right, autophagy. I can never pronounce that. Autophagy is the process by which your body actually consumes and eats up the proteins that get made, and that's broken in some way with respect to Parkinson's disease. There is evidence that suggests that mitochondria might be implicated in this kind of process. There's evidence that the process of inflammation within the brain actually have a role to play within the causes of Parkinson's disease, but we don't exactly know how. And of course, we do know that at the level of brain regions and cell populations, there's a specific set of neurons that are damaged within Parkinson's patients that causes movement disorders and other sorts of long-term devastating, horrifying effects that we want to try and understand and we want to try and stop. Okay, so in order for us to go further, we have to start building representations and knowledge engineering approaches of these various different aspects of these different research fields. And we have to find connections between them. We have to try and synthesize their knowledge so that we can actually understand and cure this disease. That's kind of what we're up to in the longer term. So coming here, I was wondering, how would Google kind of tackle this? I thought, my question was, if we were going to go Google on this problem, what would that entail? What might that look like? And so, forgive me for being a bit fanciful, but I thought, well, wouldn't it be cool if we had a supermassive repository of all the scientific data in the world? Like we found a place where we could shove everything, and we put all the different scientific observations from every single scientific experiment that has ever been done, and we put it in there. First of all, the question would be, how would we populate it? Well, since this is Google, you guys would develop really beautiful data management and analysis tools that would deliver scientific expertise to the scientists working in the lab in a way that would transform the subject. So I was thinking about this. It's like, I remember the days when I was driving around LA with my Thomas guide on my lap, and not that I ever did that, of course, but the experience of doing that, as opposed to the experience of using Google Maps to kind of examine how one gets from A to B, was transformative, right? Perhaps maybe there's something that we can do within a scientist's experience of doing work within a lab that would be analogous. That would be cool. Of course, there's a lot of data in databases already. Perhaps, you guys can figure out ways of doing deep information integration, sucking information from these open access databases. Anyone can do anything with the data from these things. Maybe there's some interesting and really fantastic ways in which we can pull data from them. And of course, the scientific literature, which is, effectively a 16th century artifact, right? These things are PDF files. They're accessible electronically, but it's still just text on a page. Now, maybe there's ways in which we can transform the way in which these things get written. Maybe there's ways in which we can pull the information out more aggressive, more interestingly, more powerfully. There's all sorts of things that we could do there. And then, of course, once we've gotten this system, what would then be possible? Well, you could do terminology analysis. You could actually develop ontologies that reflect scientists' use, so that scientists would actually be willing to use the terminologies and use these standardized terms in an easy and straightforward, very natural way. Perhaps there's ways in which we can do meta-analyses across studies, which still remains a very, very difficult and unsolved problem in general for scientists to do. Trying to actually get the data from one paper and compare it to the data from another paper and being able to make the connections between them, not an easy problem. With this kind of repository, that would be easy, that'd be straightforward. Then imagine, for example, we could scale up our representation of data massively. So if you wanted to do systems-wide, large-scale systems biology analyses of all the proteins involved in a specific disease, bang, you'd be able to do it in no time at all. And then I'm kind of excited about this idea. Judea Pearl is a giant in the field. He's someone who developed probabilistic graphical models and really made a huge difference. Now, some of his work is based upon the idea of representing causality in a computer, being able to actually look for causal relationships within the structures that he's developed. Now, maybe we could use that and we could leverage that in studying how things really work. Maybe there's actually some underlying long-term kind of interesting research that we could really leverage there. And then, of course, we could build a breakthrough machine. Okay, I don't really know what that looks like, but that's kind of what I'm up to. That's what we're trying to do. Maybe there's a way in which we can automate the process of doing scientific breakthroughs, and we can actually turn that into an engineering problem. Okay, so in that vein, I'm one of the organizers of this workshop, the Discovery Informatics workshop. Well, there's a workshop at the AAAI seminar, at the AAAI Fall Symposium series coming up in November. Deadlines for this are in 1 month. We're very, very excited to try and get people involved and interested. And we're also looking for people who might want to act as keynote speakers as well. So if there are people who could be potential speakers for this, we would be very interested in talking to you. And we're kind of excited about this whole thing. So just to summarize, I've gone through a lot of stuff and I've talked quite quickly, but I just wanted to kind of summarize, wrap it all up in both of you guys. So first of all, I've talked about conceptual developments in terms of this cycle of thinking about the scientific process as a cycle. And we want to try and represent and think about paradigms explicitly. And this is, I think, an important breakthrough that no one really is doing in an explicit way. We have our own methodology, knowledge engineering from experimental design, which is the whole way in which we can accurately capture data from scientific papers and scientific studies in the way that they were originally intended to be. We talked a little bit about topic models of the scientific literature as a methodology for identifying and examining paradigms. There's also obviously other types of information extraction work and all sorts of natural language processing that can be done in this realm that I think are very interesting avenues of research. We want to try and advocate open standards to help alleviate the problem of dark data. And then finally, I'm proposing this specific project as a target kind of goal for actually trying to address and support the Parkinson's research community in their work. This is thanks to a lot of people. I have a lot of people to thank. This is just a small subsample. Thank you for your attention. Here's my email address, my website, and my blog. And feel free to get in contact with me if you have any questions or if you have any ideas. Thank you.
We have time for a few questions that people have.
I can repeat the question too, if you like.
Is there any more low-hanging fruit with citations and papers? Usually goes to the granularity of one paper citing another.
Yes.
Typically, within a given paragraph, you'll have several citations. So like in a nano publication, you did nothing but dissect all papers into paragraphs. Has a kind of distance metric physically between citations. Is any work being done in that?
Yeah, absolutely. So Marty Hurst at, I think at Stanford, is actually developing, she came up with the notion of a citation sentence as being a kind of unit of representation that you can do this kind of work with. I'm not sure if she's specifically looking at the kind of questions that you've just described, but I think it's kind of in her wheelhouse. There's also an ontology of citations called Cito, being developed by David Shattuck at Oxford. And he's trying to provide a language for describing the different roles of a citation when one is supporting another. But what you're saying is specifically just calculating the distance metrics within the text as a way of looking at these things. I haven't heard that before. I think that's an interesting idea.
Like, one analogy is just with, say, Google search, where there's, if you do nothing more than just consider it a new metric, like distance, physical distance between words, something might appear. Or the two basic tensions are bag of words concept versus a knowledge graph, and it seems as though the citation graph seems to be traditionally associated with some kind of knowledge graph, when actually maybe more power is simply in the physical proximity of citations. And I was wondering if that was being, and that if you want to scale, that's an issue where scaling is much easier.
Yeah, I haven't heard about the physical location, but I think that's an interesting idea. I think personally, the way in which citations are used is really undistinguished. I mean, obviously you just point to a paper and you have some underlying theory of why it is you're pointing to there, but there's no way of really telling why or what the purpose of that is. So I think, yeah, I mean, and also the other aspect of this is that doing text mining of a full text is actually difficult because of licensing concerns. So we have to be able to get the text. We have to be able to extract the text from the documents that it's contained within. We have to be able to recognize citation sentences, and then we can actually start addressing the kind of questions here. I don't feel as though I've really effectively answered your question, but
That's good, fully formed.
okay, sorry.
But I will consider it more and maybe contact you later.
Sure, please do. Yeah.
Okay, so I understand that the diagrams that you show, the process model, can also serve as the description for data.
Yes.
So there is the protocol and there is inscription. So how basically how detailed do you think can be this description? So the data which you propose to collect in a big database is actually depends on the instrument or the method that it was taken, and so on and so on and so on. So increasingly we're taking data which make actually no sense without a computer from so many different complex instruments,
Right.
that you cannot look at them or something, you need to transport them.
Right.
So do you think it's how do you think it would be it would capture the description of the method and instrument and everything to use that data later, like in 5 years or 10 years when the instrument is not around?
Right. That's a good question. So I think that one size does not fit all, and that you have to tailor the representation of the data that you're capturing in a way that's appropriate for the task that you need the data at that time. Because frankly, one of the problems that we have is that, you know, the process of knowledge acquisition from these things is a bit of a rabbit hole. You can go down as deep as you like into the details of things, and unless it's going to be useful for you in the immediate future, you're not really going to be able to get people to do that. So my answer is that I don't know what level you have to take it at, but I think you want to aim for a kind of process of just getting just enough as the way of doing this. Because if you don't do that, if you dive down into as deep as you possibly can get, then the process of curating the information becomes pejoratively difficult, and no one will do it. So you have to kind of tailor your approach, and of course what you need as well is a kind of multi-level representation of the workflow. So in fact, for example, take an example. Let's say that we're doing a genetic sequencing process. You could put that in a single node in the process graph, as we've described, or you could explode that into all the different sub-steps that you need to take within that process. So from our perspective, the representation should be able to say, okay, this single process is actually made up of all these sub-processes, and have it linked together, so that then you could in theory curate both. First, when you first make the analysis, you want to curate it to the maximum of your ability at that time. But then let's say that you wanted to drill down into the data later on, you would want to explode out that process into the different subsections and do that. So I don't really have a good answer to that, because, but in theory, I mean, in theory, if you're automating this process and you're automating the process of data acquisition, just shove it all, you want to capture everything, right? I mean, that would be the ideal process. But then you'd need a representation that allows you to make sense of that, and as you say, 10 years down the line.
Thank you.
You're welcome.
Actually, there are processes which generate more data than we can capture, like, for example, the large, the collider,
Right.
which generates like 60 gigabytes per second of raw data. So they kind of process and discard them
Right.
immediately. So yeah,
Right.
we should select the right level.
Right. But there are some experiments that you can't repeat. So in those experiments, you have to try and capture as much data as you possibly can, because it's incredibly valuable to be able to do so. So, you know, that's a very important question, and something that I think scientists in the future are going to have to deal with. Yeah.
Quick question from VC. Can you hear
Go
me?
ahead. Yes, please.
So you described, representing the data acquisition process. It sounded like essentially a manual effort to do this, to create these flow diagrams, and you mentioned it could be multi-scale and so on. So once you've created this by hand, then what? Do you have algorithms that process this graph representation that you've created? Or does it somehow add, in what way does it add value to the raw data?
Okay, so we're currently in the so all of this is development work, and we have, okay, so at the moment, what we've done is we were able to actually capture the organization of the protocol, and we're trying to develop the underlying infrastructure for being able to represent data at each stage. It's still very nascent. We're still trying to look for funding to be able to develop this further and make this work more coherently. The way I think about this in the future, so this is, again, a future direction kind of argument, is that imagine, for example, that the processes that we're dealing with are kind of like a grammar. So you could, a compositional grammar to build experimental designs for a whole class of different experiments. And so the kind of underlying theoretical model that I have in my mind about how we can then process this data is that you can look at the different kind of fragments of an experiment that are dealing with measuring specific things and see how they combine, which is essentially the kind of underlying, this is the reason why this is a complicated problem and that normal data modeling methods don't work, because each experimental design, when taken as a whole, involves composing different pieces of the same underlying design, right? And so, if we are able to construct a grammar-based approach to represent this type of data, then perhaps we can use that to analyze the underlying data and the underlying structures. And then maybe I have a bunch of graphs of work that we've done, for example, in automating the comparison of different studies in studies of HIV vaccines. And so, for example, instead of a layout, the kind of graph-based layout of papers based upon the similarities in topics, we could, for example, drive analysis and a visualization of the different types of experiments because of similarities in their underlying design. So that's one aspect of how I think this kind of work could be used to help evaluate and understand the data within a field. But it is still very early days, and we're still looking to try and develop the ideas, basically.
Okay, makes sense. Thanks.
Thank you. Any other questions? Go ahead.
Some information like the axes on graphs and things like that to validate one of your models.
Yes.
How much of that kind of stuff do you think can be pulled out of papers? Like if you look at all the papers in a particular topic cluster and look at all the graphs and tables in that, just do simple analysis like that. Do you think how much of your model do you think you could potentially reverse engineer?
I think that's a really interesting question. We did some work on looking at a specific class of experiments called track tracing experiments, which study connections in the brain. And we found good results when we knew what the independent and the dependent variables were. So if you look at the results sections of papers and you have a predefined idea of what it is, the measurements that are needed to be used in this type of experiment. So for example, labeling in brain tissue, or the location of an injection, or that kind of, or a specific type of chemical that's being used within the protocol. That boils down to a simple information extraction problem. So you get the same kind of performance that you have when you're trying to pull out names of companies, for example. It's the same kind of process. So we think that we can do that quite efficiently. But we need to have the underlying models defined for for a specific experimental type. And of course, that's an interesting question because some experimental types are much more complicated than others. For example, I mean, I've just been, so the work that I've started looking at cell biology studies of cellular processes to try and get at ways in which we can look at, for example, how LRRK2 is influencing different aspects of cell biology as part of the kind of trying to study the causes of Parkinson's disease. Now, their representation of experiments in cell biology is completely different from the kind of thing that you see in vaccine studies. Vaccine studies, you basically run an experiment over you have a very complex design for a single process. Whereas in cell biology, you have 25 separate experiments, all very simple ones, where you basically get some cells, you incubate them, you run a gel, you see what it looks like. You then do the same thing again by tweaking some of the parameters, and so it's interesting and it's encouraging because we think the K-Fed modeling approach could easily handle that without too much difficulty. So you have a lightweight framework for capturing those kinds of experiments. But again, if you're trying to model if you're trying to automate the process of pulling that information from the literature, it's a whole different kind of ball of wax. You then have to identify experiment boundaries within the paper. You have to look for different things. So I think it really but that would also come down to how you prepare the background knowledge of what experiment types you're looking for and how you would use that to seed the process of developing an information extraction engine across the paper. But I think that what's interesting about that is that looking for the variables and the measurements is a really powerful